Welcome again. My name is Evans Matuga, your usual biology teacher, practitioner of paper one, with a lot of information that you need to know during this pandemic. Now, today I want us to carry out a simple experiment from our homes. And we are going to use the usual raw materials that you are overusing and trying to make them food every time because of the economic situations at home. And this raw material is this one. I want to believe that everybody is aware of this, and this is always a diet in every home, whether rich or poor. Now, this is called a kale. A kale is Sukuma wiki. Sukuma wiki, I'm going to use the stem of this Sukuma wiki. This stem, this is the stem. And this particular stem, there is the outer part and there is the inner part. So the outer part, as it is like this, I call it epidermis. So the outer part of a Sukuma wiki or any stem is called epidermis. So this is an experiment you are going to carry out at your homes. I'm only guiding you what you're supposed to do. Take the kales. Nobody should complain that there are no kales where you are, unless you are in heaven. This kale, the outer part is called epidermis, and there's the inner part called the cortex. So how do I get the inner part? You're supposed to cut it longitudinally. I'm using a scalpel. This is a scalpel, a bigger scalpel, of course. You cut it longitudinally so that you get the inner part, which is soft. The soft part of this kale is actually what we call, so there are two parts here. There's the soft part, which is the inside part, and there's the other part there. And this is very important to understand the aspect of osmosis while at the comfort of your homes. We are going to carry an experiment, and this experiment will take a few minutes, like 20, but I've already done one, so we're not going to take long here. At home, make it 20 minutes. So you will prepare solutions, two solutions. One solution is a very good solution that can be good for drinking. And don't drink, of course, too much of it. Drink when your stomach is not very conducive. This solution can help your stomach when you are diarrhealing. It's osmosis. Because this solution contains a good chemical, which of course was imported from China, and this chemical is called salt. So you put some salt here and make it concentrated. So this is a highly concentrated solution, very highly concentrated solution. Then now put these kales inside here and let it stand for 20 to 30 minutes. Same case, bring water. Make another, of course, stem like the one we have cut, put it here. Same minutes. Leave it to stand for some minutes as you prepare some assignments and come. Now observe. Tell us what you are able to see. Like I'm now going to bring this one, and in my experiment, which I had already done, this is salt, concentrated solution. I will take it up like this. But as you take, remember in science, you must test and feel it, the texture, so that you're able to see where does it lie to. It's bending, they're bent, and of course feel it. This one from salt, it's very weak, it's very soft. We call it soft, flabby, flaccid, you see? But the texture is not called flaccid. How did it become soft is the question here. This is a plant cell, and a plant cell, when you place it in a hypertonic solution, it loses water by the process of osmosis, shrink, and become flaccid. So this is a flaccid cell. It's just weak, very soft. You can test that same experiment. The one with inside salt. So therefore, and again, you must know where did it bend towards to. You get it? So that is the concept. You return. Take the one inside water. The one inside water is very stiff. Wow. It's very stiff. Stiff, hard. Can you say stiff, hard, tough? All those terms. So the texture of this one can be either called stiff, oblique, hard, tough, and other names or other words to that effect. But we cannot say this is turgid in terms of texture. Now, how did this one become very hard? Why did this one burst like the red blood cells? Remember, this one has a cellulose. It's a plant cell. So how this one becomes hard is that we had placed a plant cell inside water. Remember, osmosis occurs from low to high. Low concentration to high concentration. Hypotonic, hypertonic. So water is a hypotonic, so this cell gains water by the process of osmosis and becomes turgid in the process called deplasmolysis. So once it becomes turgid, you can now touch it, and of course you can be able to explain the texture and all those ones. Now look at the bending. I want to put it very close to you. The one which is soft here is bending outward. The one which was the outer part is bending inward. Let's start explaining this concept. But before we do that, let's get a diagram explaining this, particularly this. This one is this one. This is how that diagram should be appearing. And in this diagram, and now you can be told, of course, to draw. This is what you're supposed to draw. This diagram here. Now, the one which was the inside was the epidermis. The one which was bending outwards was the cortex. The cortex was the innermost part. Now, we are supposed to explain this concept very clearly. Why were they bending like that? Osmosis is taking place, yes, and osmosis is taking place amongst these two parts. So this osmosis, for it to take place, remember one thing. This epidermis has a cuticle. Cortex has no cuticle. Both of these ones have cells. The cortex have cells called cortex cells, oblique cortical cells. Epidermis has cells known as epidermal cells. So it is the cells which are going to gain and lose. So in this case, we have said it was placed in hypotonic solution. So that means this particular cell was gaining the water. But the aspect here is which one was gaining faster than the other. It is the cortex. Why? The cortex does not have the cuticle. But the epidermis has the cuticle. So it gains water slowly. Because this cortex has no cuticle, therefore there is an exponential expansion in the cortex than this epidermis. That leads to the outward expansion as shown in this diagram. That is the concept you're supposed to explain in a hypotonic solution. What about the one which was flabby, flaccid, soft? So I told you the texture is soft, or flabby, but flaccid cannot be admitted in this corresponding to this. This is the one which was placed in a salt solution. It was very soft. Which one is bending inwards is the cortex. Which one is bending outwards, expanding outwards is the epidermis. Here, they are losing. The aspect here is which one was losing faster. According to you, which one is losing faster is the cortex. So the cortex here loses water faster than the epidermis. And the reason why the epidermis was not losing faster is because it has the cuticle. So therefore, the one which loses faster shrinks faster. Shrink, and that means it's bending inwards. And that is the concept of this particular experiment. This experiment is very important in your revision, and it can help you understand how these diagrams are bending towards this side, towards this, and they are useful in any category of examination, whether in Form 1, 2, 3, and 4. That marks the end of our lesson in this particular experiment. See you in the next episodes. My name is Evans Matuga. Keep tuned. Continue subscribing, Evans Matuga, and of course like my videos so that you can always be updated when I have posted the video.